Okay, class, this is going to be a short video on capillary exchange. This is one of the topics that, while not necessarily the most important concept out of chapter 20, there's definitely a lot in chapter 20. It's one of the ones that can be most complicated, but hopefully after this video, you'll see that it's pretty straightforward, and it really is a fascinating physiological concept, and really see an amazing design of our cardiovascular system here and that relationship between anatomy and physiology, and especially working with what we talked about in the heart chapter, which is this idea of maintaining tissue perfusion. So as I said, this topic is capillary exchange, which ultimately answers the question and explains how material moves between the blood and the tissues. So functionally, when you look at your blood vessels, arteries send blood away from the heart, veins bring blood back to the heart, and capillaries are those tiniest little middle vessels. So they're in between the arteries and the veins. They're neither bringing it away nor to the heart. Their goal is to exchange. So material, oxygen, carbon dioxide, waste, nutrients diffuses between the blood and the tissues at the capillary. So that is the unique function of the capillary. So you see, it explains how material moves between the blood and the tissues, because this is the functional purpose. Tissues need to obtain oxygen and nutrients, and they need to get rid of carbon dioxide and waste. So tissues are where your cells are located. The cells obviously are the workhorses of your body. They are making the ATP, they are producing the proteins, they are doing the building blocks, they are doing everything, little construction workers of your body. They're located in the tissues, so the tissues need oxygen and nutrients to feed those cells, and it also needs to clear out the waste and the carbon dioxide. So before we even get into my analogy and really looking at the physiology of capillary exchange, we need to make sure we establish some fundamental terminology. So I can tell you, if you don't understand these four terms, these four definitions, you're going to have a really hard time. It's going to be really confusing reading through this physiology. So you really need to make sure you have these definitions first. So the first thing we need to know is, what is filtration? Filtration is the movement of these materials, whatever it is, movement of material out of the blood and into the tissues. So realize, even during this lecture, if I say filtration is movement out of the blood, you must know that that implies that it's moving into the tissues, and the reverse. If I say that this material is moving into the tissues, you need to understand that means by default it's also moving out of the blood. So filtration, whatever the material is, it's moving out of the blood into the tissues. Reabsorption or absorption, different books will use different terms. So you can either say reabsorption or absorption. So reabsorption is just the opposite. The material is moving out of the tissues and into the blood. Then the next pair of terms you need to be familiar with is hydrostatic pressure and colloid osmotic pressure. So first, hydrostatic, let's break the word down. Hydro means water. Static means stationary, what is latent, innate to the system. And of course, it is a pressure done by hydro, which is water. So hydrostatic pressure is the innate pressure from water pushing out from a vessel against the vessel walls. So if you think about like a water hose, the reason your water hose goes from flat and fills up is because of hydrostatic pressure. There's water inside the vessel and it's pushing out because it wants to go out. So hydrostatic pressure is a pushing away force. So I say pushing away just to be in general that whatever location we're talking about, hydrostatic pressure wants the water and the material to move away. Now, in our body, there are two locations. There's the blood/capillary. So if we want to say blood hydrostatic or capillary hydrostatic pressure, we've already talked about systemic blood pressure, same thing. But here we're talking about in the capillary. So one location is the blood capillary, or it's the fluid that's in the interstitial space, which is the tissue space. Interstitial means the tissue. Then we have the colloid osmotic pressure. So first, osmotic pressure, first this colloid osmotic pressure is a pulling towards force. So if hydrostatic pressure is, say, water pushing away from a location, colloid osmotic pressure is a pulling towards that location. So what is osmotic pressure? This is just referring to the fact that if you remember all the way probably in your biology class, osmosis, the fundamental concept of osmosis is water always moves to the area of the higher solute concentration. I like to simplify it and say water follows salt. Water always wants to move to the saltier area. It always wants to reinforce where water is less. It wants to even out. So osmotic pressure is basically a measurement of how forceful does water want to move to that salty area? How forceful does water want to move to that area of high solute concentration? In this case, I'm telling you what the solute is. Colloid osmotic pressure. Colloid means protein. So colloid osmotic pressure is a pulling towards force. And who's doing the pulling? Who is the attraction here? It's the proteins found in that fluid. That's what colloid means. So colloid osmotic pressure is a pulling towards force. It's the force of water wanting to move to the area of proteins. So hydrostatic is a pushing away. Colloid osmotic pressure is a pulling towards. And so with colloid osmotic pressure, you have a blood capillary colloid osmotic pressure, and you have an interstitial colloid osmotic pressure. So the same two locations, just a different force. So when we put all of these terms together, you get something which is kind of the end product here. This is the end goal of this lecture, is calculating and understanding the net filtration pressure, NFP. So net filtration pressure is the sum of all of the hydrostatic and all of the osmotic forces. So net filtration pressure is the end relationship between all of the pushing and all of the pulling forces. So it's the sum of the capillary hydrostatic pressure, the interstitial hydrostatic pressure, the blood colloid osmotic pressure, and the interstitial colloid osmotic pressure. The reason this is important is because the resulting number, if we put numbers to this, when you complete the equation, the result determines the direction of fluid movement. So ultimately, when you put all of this together, is fluid ultimately being filtered out of the blood, or is it ultimately being reabsorbed back into the blood? So make sure you take a moment to really wrap your head around these terms before we move into the analogy. So feel free to pause the video if you need to, rewatch it. Just make sure you have an understanding of these terms. So let's first look at what's called the net hydrostatic pressure. So the net hydrostatic pressure analogy. Remember, hydrostatic pressure is a pushing force. So if we go back here, hydrostatic pressure is a pushing away force, but remember, you have two locations that you are pushing away from. You're pushing away from the capillary and you're pushing away from the interstitial. So the net just means what is the combination of those. So let's simplify it. Let's say that you have two people pushing on this cinder block. So we have what I'm calling a right pushing force and we have a left pushing force. So how would you calculate which direction the block moves? Or let me say it this way, what will determine the direction that the cinder block will move? So think about your answer for that. Okay, well, obviously the block is going to move in the direction of who pushes the hardest. So if the right pushing force is a 10 and the left pushing force is a 5, 10 minus 5 results in a positive 5. So that means the cinder block is going to move at a force of 5 to the right because the right pusher was stronger than the left pusher. So that's how we would calculate, in this case, what's called the net hydrostatic pressure, or let's just, for the analogy's sake, let's just call it the net pushing pressure, because you have a right pushing force and you have a left pushing force. When you sum those together and you get the result, it is a net pushing pressure or a net pushing force. Okay, let's look at the other one. Let's look at the colloid osmotic pressure analogy. Remember, colloid osmotic pressure is a pulling towards force, so we can think of this as the pulling towards force, or pulling towards pressure analogy. So we have a left pulling force and we have a right pulling force. How would you calculate which direction the pulling the block moves in this case? What will determine the direction that it moves? Okay, well, same thing. Who is going to pull the hardest? So let's say that our left puller is pulling with a force of 10, and our right puller is pulling with a force of 5. Well, once again, we have 10 minus 5 is a positive 5. We know he was the strongest at 10, so then the cinder block is going to get pulled, in this case, to the left, towards the direction of the stronger puller. So that gives you a net colloid osmotic pressure. So we have the net hydrostatic pressure, and this would be the net colloid osmotic pressure. So you can see that we had net hydrostatic pressure, or the net pushing pressure. We have the net pulling pressure, or the colloid osmotic pressure. But the goal is to put all of this together. So let's do a little bit of math here. What you just did, now let's actually put some numbers to it. So for this first step, so we have both pushers and we have both pullers. So this right here would be the net pushing pressure. This right here is the net pulling pressure. So we want to figure out the net movement of the cinder block now with all 4 forces added in. So for this first question, put them all together. So we're putting all of these values together to determine the net direction of movement. So given the following force values, determine what will happen to the block. So right pushing is 35, left pushing is 0, left pulling is 26, and right pulling is 0. So take a moment, pause the video if you need to, and figure out what is the overall net direction of block movement. Which direction will the block move? How can you figure that out? Okay, hopefully you've had a chance to figure this out. Like I said, pause the video if not. Well, let's do just what we did, break it down into those 2 steps. So let's look at the net pushing pressure. So 35 minus 0 is 35 to the right. Let's do the net pulling pressure. 26 minus 0 is 20, results in a value of 26 to the left. So we have a force of 35 to the right, a net force of 35, pushing force of 35 to the right, and a net pulling force of 26 to the left. So now we're pushing, let's think of it this way, if you're pushing 35 feet to the right, and then you're pulling back 26 feet. So what is going to be the difference there? It is a value of 9, a positive 9. And with the way that we set up the equation, that means the cinder block will move, in this case, 9 feet to the right. We pushed it 35 feet to the right. We pulled it back to the left 26 feet. So that means the net change ended up being 9 feet to the right. So I hope you followed that. If not, rewatch the video, rewatch the steps that we got there. We just did it in 2 different steps. So we figured out the net pushing force, the net pulling force, then we put everything together. So now let me change up the values. A very minor change here, but try it this time. Put them all together to determine the net direction of movement. Given the following force values, determine what will happen to the block. You can see our change here is that instead of 35, it is now 16 to the right and 26 to the left. Okay, same thing. So we do 16 to the right. So 16, so net 16 to the right, and then we're back 26 feet. So what is the net change here in block movement? 16 minus 26, in this case, is a negative 10. So we went 16 feet to the right, but then we get pulled back 26 feet. So we actually end up now a net change of 10 to the left. Here we were 9 to the right, now we are 10 to the left. So you can see just by changing one of the values, and of course these are 0 over here, but if we change those numbers, we just still work the equation the same way. That gives you that net change in cinder block movement. So once again, with our analogy, we just need to remember that we have pushing forces and we have pulling forces, and we're trying to figure out, and we're calculating the net change of each of those. So let's make it actually real. Let's take our analogy and apply it into your body, into your capillaries. So instead of saying that we have a right pushing and a right pulling and so on, let's actually put some terminology to this, and we've already been introduced to these terms. So the right pushing is actually what I'm calling, the right pushing is going to be labeled the blood capillary hydrostatic pressure, obviously is a pushing force. The left pushing is the interstitial fluid hydrostatic pressure. The left pulling is the blood colloid osmotic pressure, and the right pulling is the interstitial fluid osmotic pressure. So all we did was just change the names, but we can see that the analogy holds true whether we're talking about right or left pushing or right or left pulling. And you can look at this in your book. But for those of you who like to see the math equation labeled out, I have it here, and obviously you can pause the video and look over this. I'm not going to explain every terminology, every detail in here, but ultimately all we've done up to this point, and this is just the summary. So if we want to determine the direction of fluid movement, which is the net filtration pressure, we need to calculate the net hydrostatic pressure, and we need to calculate the net colloid osmotic pressure. So if we break that apart, we get NFP equals blood colloid hydrostatic pressure minus the interstitial fluid hydrostatic pressure. So we calculate that. And then we're going to subtract from that. We're going to have that combination here from the blood colloid osmotic pressure minus the interstitial colloid osmotic pressure. So we're calculating the net pushing force, and then we're calculating the net pulling force. And so you can see your book, depending on what book you have, may use slightly different terminology or slightly different abbreviations, but this is what I think is the most straightforward. And so you can see the key here, and all of these terms are things that we've looked at. And you can see with the interstitial fluid hydrostatic pressure and the interstitial colloid osmotic pressure, these values are usually 0, or they can be 0. So just for the sake of simplicity for the application, I'm going to assume that they are 0. So here's the takeaway. If after you've done this equation, which we've already done, remember, we've already figured this out just by doing these calculations. We did the net pushing, we did the net pulling, and then we figured out the difference. We did the net pushing, we did the net pulling, we figured out the difference. If your number, if you set it up in this way, if your number is positive, then it means fluid was filtered. And remember, filtered is a pushing out. So ultimately, the fluid was moving from the blood into the tissue. If the NFP is a negative number, then fluid is reabsorbed, moving from the tissues back into the blood. The reason this is important is because these values are going to change at the arterial end of the capillary versus the venous end of the capillary, which is exactly why I gave you 2 different examples here, 35 and 26, and then 16 and 26. So this is the figure. Sorry, it's a little fuzzy here in the PowerPoint, but if we turn our cinder block on the side, we can apply it to the image, kind of make it look similar to the image that you have in the McKinley book on capillaries. Like I said, this is kind of fuzzy. This is page 799. But for the most part, the values are going to be the same. One of the differences here is you can see 35 and 26, they're 16 and 26. Those are the values that I used. Let's see here. So we have 35 and 26, and we have, oops. And we have 16 and 26. The reason that that the ultimate result of each of these numbers is going to change is because you can see that in your book they're a little more accurate, that they actually calculate the let's see, what do they call it? Just to see the net colloid osmotic pressure of the interstitial fluid is they use a value of 5, which I did not do in our simple analogy, just to keep it straightforward. But that's why these values 14 and -5 are going to be a little different. But of course, the process of calculating it is exactly the same. So 35 and 26, 16 and 26. And then if you look in your book, you can see how they do the equation, adding in that -5. But the end result of all of this is going to be on the arterial end, when you do the equation, is a positive 14. And when you do these numbers this way, is a -5. Now, the number itself, 14 or 5, I don't really care. That number itself is relatively arbitrary. It's hard to get accurate numbers on this. But what I want you to know, the takeaway here is that when you do the equation, the application of the equation, the understanding of the values here is that at the arterial end, it's a positive 14. It's a positive number. We just learned on the previous slide that a positive number means it is being filtered out of the blood into the tissue. And then at the venous end, it's a -5. So negative means that fluid is being reabsorbed from the tissue back into the blood or back into the capillary. And so this changes, it slowly decreases from positive 14 all the way to -5 as you move along the capillary. So what's interesting is that you can see here, if we use these numbers 14 and 5, it means more fluid is filtered out at the arterial end than is actually returned at the venous end. So let's do a little summary here. So fluid exchange homeostasis. Notice that more water is filtered out of the capillary than is reabsorbed back into the capillary. Well, what do you call it when fluid builds up in your tissue space? That's called edema. Well, obviously edema is swelling. That's bad. And assuming you are healthy, you can look at your body and you notice there isn't swelling. Roughly 2 to 4 liters a day of fluid leaves your capillaries, enters the tissues, and does not return into the capillary. So this is actually the role of the lymphatic system that we're going to get into in a chapter or two. Is the lymphatic system helps to return this fluid back into your bloodstream. So when we look overall at fluid exchange homeostasis, when we go and look at those basic terms, when we look at hydrostatic pressure, when we look at colloid osmotic pressure, remember hydrostatic pressure is related to blood volume, to the water level, and the colloid osmotic pressure is related to the amount of solute or protein that's in your blood. So determining either we're pushing or we're pulling and the amount. So you can think through these. These are the application questions here. How will the net pressure, so how will the net filtration pressure change? How will the net colloid osmotic pressure change? And ultimately, how will the net filtration pressure, how will the ultimate NFP change if you are dehydrated? So think through what happens when you're getting dehydrated. Your body is losing water, which means your blood is becoming thicker, it's becoming more solute, it's becoming more salty, it's becoming more protein. So what will that do to the direction of fluid movement if, in this case, the blood colloid osmotic pressure is increasing? Well, now what happens if you drink too much water? What will happen if your blood volume goes up? Or what will happen if your solute concentration goes down? What will happen if your liver doesn't produce enough plasma proteins? So let's say that you have liver damage, like cirrhosis of the liver, some sort of problem. The liver's not producing enough plasma proteins. Is that going to primarily affect hydrostatic pressure or blood colloid osmotic pressure? Or what happens if you are hypertensive, if your blood pressure goes up? How is that going to influence these individual values? So think through these. You have your homework questions that will relate to this, obviously from your book, and feel free to reach out if you have any questions for me, and I'll be happy to lecture on this further in a one-to-one Zoom.